Let's move ahead. So everything has a basic structural and functional unit. As your functional unit is cell, so the DNA's functional unit are nucleotides, right? So nucleotides are basically of two types, that is purine and pyrimidines. What are basically nucleotides? Nucleotides are composed of three things. One is a pentose sugar, the second one thing is nitrogenous bases, and the third one thing is a phosphate group. So these three things combine together to form a nucleotide. If a phosphate group is removed from these three, and only there are two things like pentose sugar and nitrogenous base, it will be called as nucleoside. Right? So you can say a phosphate group attached to the nucleoside becomes what? Nucleotides. So there are two types, purines and pyrimidines. What are basically purines? Purines are, there are two types of purine, adenine, and second one is guanine, right? Adenine and guanine, you can memorize it as a pure as gold. P is for purine, pure purine, as A is for adenine, and G of the gold for guanine. This mnemonic is written on the first aid book. Let's move toward the pyrimidines first. So there are three types of pyrimidines. The first one is cytosine. The second one is thymine. And the third one is uracil in case of RNA. In case of DNA, there is only cytosine and thymine. And thymine is replaced by the uracil in RNA. So what happens? You can say that just methylation is required, only a methylation is required, methyl group is required, methylation is required for uracil to become thymine. That is in the case of DNA. So you can memorize it by a mnemonic, cut the pyramid, cut, C-U-T, cut the pyramid, right? C is for cytosine, U is for uracil, T is for thymine, and the pyramid means pyrimidines, right? You can memorize it by this way. So what basically happens, the purine combines with the pyrimidine in a way that always adenine makes a base pair with the thymine, right? And guanine makes a pair with the cytosine, A, T, C, G. So I can write it here, A with T and C with G. There are two bonds between adenine and thymine and three bonds between cytosine and guanine. So as you can see that C and G has three bonds, so it is stronger than this A and T bond. So you can say that the higher CG base pairs, the higher will be the melting point, right? And you can use this C for crazy and this G for glue, right? It's a crazy glue. It's making a glue because of three bonds. It's a stronger bond because there are three bonds between C and G, and A and T has two bonds. Now let's move toward the structures first, how they are like arranged, how the bases are arranged on the pentose sugar. Let me tell you that purines are double ringed structure. Double ring structures. And pyrimidines are single ring structures. Single ring structures. Let me draw here purine, and I'll tell you that How to memorize it? Simply, you can see C with the C, right? C with the C. One ring is like, here is N and here is also N. This N and this N are combining with the C. Now, this on this side there is a nitrogen. You can say on this side there is a carbon. On this side there is nitrogen. There is also nitrogen. This C combines with the nitrogen and this N combines with the carbon and they'll form a bond. So this double ringed structure is for purines. Now you can see there are 4 nitrogens here. 4 nitrogens here. In the next section, when we will discuss the purine and pyrimidine synthesis, you'll get to know that how these structures actually comes from. So this nitrogen is basically, this nitrogen, this one, came from glycine. This is an amino acid glycine. This nitrogen comes from an amino acid aspartate. And these 2 nitrogens came from glutamine. Glutamine. Keep it in mind that this nitrogen comes from glycine, this nitrogen comes from aspartate, and these 2 nitrogens come from glutamine. I know this is a little bit confusing in the first lecture as you are not aware of the synthesis, but in the next section we'll discuss in details how this structure comes from, right? So this structure is just a nitrogenous base. This is just a nitrogenous base and double ringed structure that is attached to a pentose sugar. And I told you that this is a pentose sugar. Let's say this is carbon number 1, 2, 3, 4, and there is carbon number 5, and to the carbon number 5 there is attached a phosphate group, and to the carbon number 1 there is attached this base, right? So a pentose sugar, a phosphate group, and a nitrogenous base will make what? Nucleotide, right? Now let's come toward the pyrimidine structure, and I told you that this is single ring structure. So you can memorize that on one end there is carbon, on the other end there is nitrogen. This carbon combines with nitrogen, this carbon combines with another carbon, this will combine with the carbon and nitrogen, and this nitrogen will combine with the carbon and they will form a bond. This is a single ring structure of pyrimidine. I know this is a little bit confusing, but when we will discuss about the purine and pyrimidine synthesis, everything will be okay. So this is all about the fundamentals of DNA. We have discussed in details. This is the first lecture which is all about this one, and now in the next section we are going to discuss the structures, how these structures actually comes from purine and pyrimidines. Thank you so much. If you like my video, please share and subscribe my channel. Thank you.